Mm, Namaskar, my dear students. Hope you are all fine. And today, in continuation with the last topics, now we have reached that we can now start the next topic, that is the second last chapter of your botany portion, and the name of the chapter is Respiration in Plants. Now, my dear students, as you know, all of us breathe to live. But why is breathing so essential to life? Have you ever thought of? Actually, all living organisms, they need energy for carrying out daily life activities. Maybe it is for absorption, transport, movement, reproduction, or even for breathing itself also we need energy. Now, have you thought where from this energy come? We know we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Now, these are several questions that we will be getting the answer in this chapter. Sometimes you may wonder at several questions. They may seem you to be disconnected, but in reality, the process of breathing is very much connected to the process of release of energy. Now, as you can see in this figure, but I have depicted here that all the energy required for the life processes is obtained by the oxidation of macromolecules to which we call food. Now here you can see only green plants and cyanobacteria can prepare their own food by the process of photosynthesis, by which they trap the energy of the sun, convert it into chemical energy, and this energy is stored in the bonds of carbohydrates like glucose, sucrose and starch in different organisms. We must remember that in green plants too, not all the cells, tissues and organs they photosynthesize, only the cells containing the chloroplast that are most often located in the superficial layers of the leaf they carry out photosynthesis. Hence, even in the green plants, all other organs, tissues, cells that are non-greens, they need food for oxidation. Hence, food has to be translocated to all the non-green plants. Animals are heterotrophic, that is, they obtain food from directly from the herbivores or indirectly as the carnivores they take the herbivores. Saprophytes like fungus, they are dependent on dead and decaying organisms. Now, what is important to recognize is that the uh, ultimately all the food that is required for the life processes comes from the photosynthesis. As you can see in this figure, this is depicting the figure of the chloroplast. You can see it is taking the energy from the sun, and this energy is trapped in the chloroplast, where it releases the oxygen as a byproduct of the photosynthesis. Glucose is prepared, which is the storage material of the food, and this glucose, when reaches the mitochondria, to which we also call the powerhouse of the cell, it undergoes the process of respiration and releases the byproducts as water and carbon dioxide, and the energy so released is used for the various life processes. To be more clear, I will show you this figure also, wherein you can see photosynthesis versus respiration. In photosynthesis, the carbon dioxide and water, they add up and make the glucose or the storage material, which on respiration that complex molecule gets broken down and the energy is released. Well, the answer to the question that whether the plants they require the oxygen for respiration to occur, they give out carbon dioxide, can be understood from this diagram. Plants have the system in the place that ensures the availability of the organisms. Now the question arises that energy is liberated in what manner? To this we can say it is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped as chemical energy, and the energy currency is the ATP, adenosine triphosphate. Hence it is important to understand that the energy released by the oxidation in the respiration is not used directly, but it is used to synthesize ATP, which is broken down whenever, wherever energy needs to be utilized. That's why it is called as the energy currency of the cell. Now we are in a position to define the term respiration, and because we are dealing with the respiration in the plants, I would define it as a process in which the complex organic substances like carbohydrates, proteins, fats are broken down to release carbon dioxide and water. The overall reaction, it is C6H12O6, that is glucose plus oxygen undergoes oxidation to release carbon dioxide, water plus energy. It requires substrate, and substrate means which will be broken down, which could be carbohydrate, which could be fat, and certain cases it could be protein. Breaking down of CC bond, carbon-carbon bond of complex compound through oxidation within the cell, leading to the release of considerable amount of energy, is called as cellular respiration, and the compounds that are oxidized, whatsoever it is called as the respiratory substrate. Now, because we are dealing with the plants, and we must know how does the respiration takes place, and I have to tell you that the overall reaction of the respiration can be given like this, that is C6H12O6, which is glucose. It reacts with oxygen. If it is the carbohydrate is the substrate in this case, energy gets released on its breaking down. As I told you that the energy is stored in the CC bond, that is energy is stored in the carbon-carbon bond, which gets released on its breaking down. Carbon dioxide is released, which goes back to the atmosphere, whereas the water is formed as a byproduct of the respiration. Now the question arises, from where does, at which place the respiration takes place? Now, since the body of the plants is mostly made up of the tissues and the cells, so the it does plants do not have a well developed respiratory system as in the animals. We have the nasal chambers, we have the tracheoles, trachea, then lungs, etc. Whereas in case of plants, we have a system of stomata, singular is stoma, plural is stomata, stomata and the lenticels across the plants to carry out the process of respiration. In stems, the living cells are arranged in thin layers inside and beneath the bark. They also have the openings to which we call lenticels. The cells in the anterior are dead and provide only mechanical support. The most of the plant cells have at least a part of their surface in contact with the air. This is facilitated by the loose packing of the parenchyma cells in the leaves, stems, roots, etc. Now, as you can see, the lenticels, they and the stomata are the two organs which make an interaction between the plant and the outside gases exchange. And how they differ? First, I will show you the diagram. This is the stomata, which I will be explaining to you just now, and this is lenticel. Now, where it is present, I will just tell you. As you can see, the stomata are located in the epidermis. They are mostly located in the epidermis of the leaves, but can be present on the other organs also. They have a special capacity that they can regulate its opening. That is, they can open and close at different intervals of time, at different times of the day, and the different physiology of the plant. They can regulate their opening and closing. The cells, the pore is called, as you can see here, this pore is called as the stoma, and the cells surrounding the stoma are called as the God cells. Now, here the two figures are seen in the figure. One is showing the stomata open and other is the stoma closed. Because of the variable turgidity of the cell, which may vary at different times depending upon the different conditions of the plant. And then the next is the transpiration. Now, what does this stomata does? It causes the transpiration and exchange of gases. This exchange of gases ultimately maintains the process of photosynthesis as well as the respiration. On the other hand, you can see the lenticels. They appear like these. These are the lens shaped, hence the name lenticels. Lens shaped structures present on the stem of the plant. Here you can see these are totally open and these are surrounded by the underlying parenchymatous cells. And what does they do? These they have no chloroplast and hence they do not take part in the process of photosynthesis. Gas exchange and little amount of water vapor is given out and they always remain open. So these are the two structures which maintain the process of respiration by bringing about the gaseous exchange between the atmosphere and the plant surface. Now let us come to the process of photosynthesis. The process of photosynthesis, it starts with the well-known process of the glycolysis. Now, all the living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called as glycolysis. The term glycolysis was originated from the Greek word glykos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Emden Meyerhof and J. Pornus. Hence, it is called as Emden Meyerhof and Pornus EMP pathway. In anaerobic organisms, it is the only process of respiration. Glycolysis occurs in the cytoplasm of the cell and it is present in all the living organisms. This is many times a question is asked, where does the process of glycolysis takes place? And you must remember, it does not take place in mitochondria. It takes place in the cytoplasm of the cells. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, glucose is derived from sucrose, which is the end product of photosynthesis, or from the storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and then two monosaccharide readily enter into glycolytic pathway or the glycolysis or the EMP pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are the same, only this step is different. As you can see in this figure, in glycolysis, a chain of 10 reaction under the control of different enzyme, because every reaction is controlled by a different enzyme and hence it is called as a enzymatically controlled reaction. It is called so various enzymes. So because these enzymes are basically the catalyst and they enhance the rate of the reaction. The Now you can see in this figure that glycolysis, we see that there is the utilization or the synthesis of ATP or NADH plus H takes place. ATP is utilized at two steps. First, in the conversion of glucose to glucose 6 phosphate, and second in the conversion of fructose 6 phosphate to fructose 1,6 biphosphate. Fructose 1,6 biphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. We find that there is one step where NADH plus H plus is formed from NAD. This is when 3-phosphoglyceraldehyde is converted to 1,3-biphosphoglycerate. Two redox equivalents are removed in the form of two hydrogen atoms from phosphoglyceraldehyde and transferred to molecules of NAD. As you can see in this figure, phosphoglyceraldehyde is oxidized with inorganic phosphate to get converted to biphosphoglyceraldehyde. The conversion of biphosphoglyceraldehyde to 3-phosphoglyceric acid is also an energy shielding process. This energy is trapped in the formation of ATP. Another ATP is synthesized during the conversion of phosphoenol pyruvate to phosphoenol pyruvic acid. Now, if you are asked to calculate how many ATPs are formed or lost during the process, can you do? Now, my dear students, you might be thinking while studying this process of glycolysis that at a number of steps you find a number of reactions, but if you go through this process once, twice and thrice, these terms will become familiar and you will remember all the names. So, pyruvic acid is then the key product of the glycolysis. This is the end product of the glycolysis, but the starting product of the next phase. There are three major ways in which the cells handle pyruvic acid. Now let us discuss them. Now, cellular respiration. You can see here what happens that there are the lactic acid formation, alcoholic fermentation, and then there is the aerobic respiration. In short, I can tell you that once the glycolysis has taken place, which is the uh, glycolysis which is a process common to both aerobic and anaerobic condition, then now it is up to the cell where it is present that it either enters the anaerobic condition or the aerobic condition. If it enters the anaerobic condition, like here, if it enters the anaerobic condition, then it can either go for the alcoholic fermentation or for the lactic acid fermentation, and if it is goes for the aerobic respiration, then it enters the Krebs cycle. We will study them one by one, all the fates which after pyruvic acid undergoes after the glycolysis. To start with, I will talk about the fermentation, alcoholic fermentation and lactic acid fermentation. In fermentation, as it occurs in yeast, you might be quite familiar with the yeast, use of yeast in our kitchens and in our bakery and industries, where the a lot of fermentation process is used for the enhancing the nutritional values of our food. The incomplete oxidation of glucose is achieved under anaerobic condition by a set of reactions where pyruvic acid is converted to carbon dioxide and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase, these are the two enzymes, they catalyze these reactions. Other organisms like some bacteria, they produce the lactic acid from pyruvic acid instead of alcoholic fermentation. As you can see in this figure, in the alcoholic fermentation, The end product is the ethanol or the alcohol. The steps involved are shown in this figure. In animals also, like muscles during exercise, when the oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase, which can also occur in the higher organism. The process is called as the fatigue. The reducing agent is NADH plus H plus, which is reoxidized to NAD in both the processes. Now, as you can see in this figure, in both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of energy in glucose is released, and not all is trapped as high energy bonds of ATP. Also, the processes are hazardous. Either acid or alcohol is produced, which is the net uh, ATPs are produced, lesser number of ATPs are produced. And now I will be telling you, I will be preparing the balance sheet for you, that how many ATPs are produced at the end of alcoholic fermentation, at the end of lactic acid fermentation, at the end of glycolysis, and we will do this when it enters the last phase of the process. Now, yeast, as you know, because either alcohol or lactic acid is produced, yeast poison themselves to death when the concentration of alcohol reaches about 13%. That is because it is a hazardous process. What then is the process by which the organisms carry out the complete oxidation of glucose? Then the complete oxidation of glucose is carried out by the aerobic respiration. Now, the aerobic respiration is the process of respiration. In eukaryotes it occurs, and these steps takes place within the mitochondria, and this requires the oxygen for its occurrence. This is called as aerobic respiration, and in this process there leads a complete oxidation of organic compounds. In presence of oxygen, there is release of carbon dioxide and water, and a large amount of energy whatsoever is present in the substrate is released. This type of respiration is most common in almost all the higher organisms. Let's have a look, how does this process takes place? This process takes place in 3 steps. The glycolysis, where glucose breaks down into 2 molecules of pyruvic acid. Of course, this process occurs in the cytoplasm and is common to both anaerobic and aerobic respiration. Then is the Krebs cycle, which is also called as the citric acid cycle, which breaks pyruvic acid to carbon dioxide, and it occurs in mitochondria and requires oxygen for its occurrence. Then last is the electron transport chain, electron transport system, ETC. It transfers that the transfers that were removed from Krebs cycle intermediates. It actually captures the energy in the form of ATP, occurs in mitochondria, and oxygen is used here. This is the passing on of the electrons removed as a part of hydrogen atom to the oxygen and simultaneous synthesis of ATP. Now, when end product of glycolysis pyruvic acid, before it enters the Krebs cycle, it has a fate which can be discussed in a separate heading. Now, diagrammatically, you can see how does the anaerobic respiration takes place. Glucose is broken down to form pyruvate. Then pyruvate forms the acetyl coenzyme A. Then occurs the citric acid cycle or the Krebs cycle. Then electron transport chain proceeds, and it is how it occurs. Now, let us discuss the fate of the pyruvic acid. Pyruvate or the pyruvic acid, which is formed by the glycolytic catabolism of the carbohydrate in the cytosol, After it enters the mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reaction catalyzed by pyruvate dehydrogenase is the name of the enzyme. The reaction catalyzed by pyruvate dehydrogenase requires the participation of several enzymes including NAD and coenzyme A. As you can see in this figure, three-step oxidation process occurs. One carbon dioxide is released, NAD is reduced to NADH and produces acetyl coenzyme A, and it is this acetyl coenzyme A which enters the Krebs cycle. Now can you think where does this carbon dioxide goes? This is the carbon dioxide that we exhale, and the plants exhale during the process of respiration. Now next is the after it has formed the acetyl coenzyme A, it enters a cyclic pathway, tricarboxylic acid cycle, more commonly called as the Krebs cycle after the name of the discoverer, its scientist Sir Hans Krebs, who was first uh, regarded to have elucidated it. Now, don't be afraid of seeing this complex series of reaction, because once we understand, they become very familiar and understandable. So let us see how does the fate proceeds in this cycle. The tricarboxylic acid cycle starts with the condensation of acetyl group with oxaloacetic acid and water to yield citric acid. You can see in this figure, this is the first step. The reaction is catalyzed by the enzyme citrate synthetase and a molecule of coenzyme A is released. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation, leading to the formation of alpha-ketoglutarate acid and succinyl coenzyme A. In the remaining steps of citric acid, succinyl coenzyme A is oxidized to oxaloacetic acid, allowing the cycle to continue. During the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is formed. GTP stands for guanosine triphosphate. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted into GDP, that is diphosphate, with the simultaneous synthesis of ATP from ADP, that is the phosphate gets shifted from guanosine to adenosine. Also, there are 3 points in the cycle where NAD is reduced to NADH plus H, and one point where FAD is reduced to FADH2. The continued oxidation of acetyl coenzyme A via the tricarboxylic acid cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. Oxaloacetic acid was the compound from where the cycle started. Now, in addition, it also requires the regeneration of NAD, FAD from NADH and FADH respectively. Now, to summarize, we can say that pyruvic acid plus 4 NAD plus FAD plus 2 water plus 2 ADP plus phosphate molecule, they in the mitochondrial matrix, they form 3 molecules of carbon dioxide, NADH, FADH2, NADH 4 molecules, 4 hydrogen molecules plus ATP are produced. We have till now seen that glucose has been broken down to release carbon dioxide and 8 molecules of NADH plus H positive. 2 molecules of FADH2 have been synthesized besides just 2 molecules of ATP. Now, this you might be wondering that neither oxygen has come out in the picture nor the promised large number of ATP have yet come. Now these all will be coming in the electron transport chain that we will be discussing in the next lecture. Till then, that's all for today. So let me summarize what we have studied today. We have talked about the process of respiration, its need, its types, that is aerobic and anaerobic. Then in anaerobic we have talked about the alcoholic fermentation, the lactic acid fermentation. Then we have talked about the aerobic respiration, in which we discussed glycolysis, then pyruvic acid conversion to acetyl coenzyme A, then Krebs cycle. Next time we shall be discussing electron transport system and oxidative phosphorylation. Till then, there is a homework for you, that you calculate how much energy compounds are released till now, so that will enable you to form the balance sheet of respiration, that how many energy molecules from one molecule of glucose are released. So we will continue this topic. Till then, thank you, stay safe and take very good care of yourself.